So our next speaker is Brenda Ogle from Minnesota, and her talk is going to be extracellular matrix-guided endothelial differentiation to support thick engineered tissues.
Thank you. Thank you very much. Thanks, as everyone else has said, but a real sincere thanks to Jay, GQ, and Joe for organizing this meeting several years in a row now. I find it a really great meeting for me. I think it's a really important opportunity for this subfield to get together, and nice that more senior investigators and emerging investigators are here to share and sort of get some momentum behind the field and also keep it growing. So in the spirit of that, I'd like to share with you some of our newer, unpublished, and really incomplete work to try to stimulate discussion, conversation, and maybe also convergence of other experiences and experiments that you might have related to this topic. My group is largely interested in the role of the extracellular matrix in supporting stem cell behaviors, especially differentiation. And so we're down here in this part of the pie. I think most of the field has been highly successful and focused in the area of secreted factors, but this area is growing. And I've always been interested in the extracellular matrix, but especially recently with the advent and growth of 3D bioprinting. I really think that the more we learn about the extracellular matrix, it's sort of an obvious bioink supplement or substance. And so that's a big part of what we're doing right now, is to try to unravel mechanisms and also just understand how best to build more complex tissues now that we have the technology. And because of this and some of the efforts that we've had going on, we collaborated with Jay's group and Paul Campanolo's group from the University of Wisconsin to actually bioprint a patch that helped with the organization of iPS-derived cardiac cell types. And I think efforts like this will continue to grow, we hope, and maybe with some of the information we're developing in the studies that I'm going to show you today. So I would say even if you're not interested in 3D bioprinting, you might also be developing some other kind of containment vessel, 3D containment vehicle maybe for your iPS-derived cardiac cell types. That's why I call it the unavoidable ECM. Either you have that or you're going to be injecting cells in vivo, and so they're going to come into contact with the ECM. Fortunately, we know that the ECM is a powerful stimulus for differentiation and maturation. And the number is sort of illustrated by the number of publications emerging in this area. So last night I did a quick search on, the search was stem cell differentiation and the extracellular matrix, excuse me, in the title. And you can see the citations sort of growing over time. And I hope that continues to grow, but I think a lot of the studies are sort of, we have this extracellular matrix protein or proteins, and we put stem cells on top of them or within them, and we saw this differentiation or functional outcome. And I think those are important studies. We certainly contributed to that body of work. On the left is some of our earlier work showing heterogeneous differentiation, in this case of human mesenchymal stem cells in response to extended culture, just on 2D films of individual ECM proteins. And I was sort of shocked at the time to see that without the addition of any other soluble factor stimulation, at least the beginnings of a differentiation process could occur. And then when we moved to 3D culture systems, we saw something similar, but in an augmented way. And the 3D in vitro model system that we were using and continue to use is based on the utilization of a 3D PEG hydrogel that's cross-linked by native chemical ligation, so it's very mild. The purpose of it is to entrap, basically, extracellular matrix proteins and cells of interest, in this case mesenchymal stem cells, human, with different individual ECM proteins or combinations of ECM proteins. And you can see by looking at a 3D rendering of the interior space, anything that's black you see is the PEG, sort of holding it in position. And the green in this case is ECM, red is cell body, and blue is nuclei. So you can see that with this system, the ECM is in close apposition and in 3D apposition with the cells. And this is hard to do, especially for things, ECM proteins like laminin, 111, 411, et cetera, fibronectin, that don't spontaneously polymerize outside the body or form hydrogels. And so using that system, we sort of began to appreciate that we were looking at mesenchymal stem cells, so we were looking at emergence of mesenchymal cell types, but we really didn't know what globally is happening with these cells when you put them into a 3D environment with the extracellular matrix. So we decided to do RNA-seq to see everything, basically, that was going on. And so what we did is we entrapped mesenchymal stem cells with either type 1 collagen, fibronectin, or laminin 111, or BSA alone. And we compared them to 2D cultures that just were at day 0, and also 2D cultures that were maintained for 28 days, like the other 3D composites, we call them. So this is a comparison of composites with BSA, fibronectin, type 1 collagen, and laminin. And if you look at the differentially expressed genes between the 2D at 28 days and the 3D model systems, and you look at pathway analysis, it's maybe not surprising that the most prevalent pathway that emerges is protein degradation. So they are in contact with the ECM, they're trying to remodel it, they've been exposed to it. But surprising to us was that development and differentiation was the second highest pathway, other than other, and none of the subcategories were bigger than that. So it's really a dominant pathway that emerges when cells, in this case mesenchymal stem cells, are exposed to the extracellular matrix. And then interestingly, if you subdivide that by, according to the type of extracellular matrix that they're exposed to, it's not just the three-dimensional construct. So that does induce some level of differentiation. You see skeletal muscle coming up with BSA alone, and then just general developmental processes. But if you look at laminin, for example, it pops up in many different tissue types. Type 1 collagen is a smaller subset, and then for fibronectin, really the only one that emerged was vascular development, which we thought was interesting. So to try to sort of clean this up, we're thinking, so what are the additional signals that are triggered when a cell comes in contact with the ECM that sort of help push it down a specific lineage? And so we had the idea, and we're still probing this data set, and I invite all of the rest of you who are sort of interested in big data handling to maybe add this data to your conglomeration. Essentially, we've honed first on, is the ECM itself evolving? So we expose it first to a controlled exogenous ECM, but then does that ECM or the remodeling that occurs actually contribute to specification? And what we saw was that maybe it's not so strong, actually. So this is a hard table to interpret, but basically on the right, and you can look in this paper now, are showing significance levels. And A through D indicate significance compared to the 2D culture, and E through H indicates significance relative to BSA in the three-dimensional construct. So we're actually most interested in those E through H letters. And they come up in a few different ECM components, and we're probing those in greater depth. But it wasn't sort of as overwhelming as we thought it might be if the ECM was evolving. Same with integrin expression. Really only integrin alpha V was different relative, or changed at least at that 28-day time point in terms of gene expression. Now I should note that it will be better, and we're trying to do this in the future to look at what's actually present, since the ECM is evolving during that could be evolving during that 28-day period, and we're not catching it by looking at gene expression at the end time point. We also looked at MMPs as another sort of function of an evolving ECM, and this was a little more interesting, and we're sort of probing this more. There's a lot of activity that emerges at this late time point, and so it could be that MMPs are contributing to degradation, and even those degradation products could be augmenting signaling that enhances specification. And so I would say that this is a data set that we're beginning to probe, and again, I sort of offer it as something that's available to others as well. Another thing that we thought of is to, instead of providing a single extracellular matrix proteins and just looking at what evolves, more of a bottom-up approach where we specifically tailor that initial exogenous ECM to try to drive specific differentiation. And I've showed this data before, but just as a reminder of what we've been doing in that same model system, and in the interest of trying to generate cardiomyocytes, we've taken different combinations of ECM components prevalent in the heart and designed an design of experiments approach to cover a parameter space, and then using statistical tools tried to hone in on a theoretical optimum, so ECM formulation that promotes cardiomyogenesis. And we did, in fact, hone in on a formulation. Again, I've showed this before and it's published, so I'm not going to dwell on it. We've been moving also to try to develop formulations for guiding endothelial specification. And I didn't think I would share any of this because we're really just the early stages. But if we entrap elements of the vascular ECM, and we've looked at fibronectin, collagen type 1 for laminin 111, 411, and 511. To our surprise, laminin 111 does give rise spontaneously. So again, this is without any other exogenous stimuli, no VEGF, nothing else. We see endothelium on the exterior surface of some of the cell aggregates, but with 411 and laminin 1, some really spectacular differentiation results, and even maybe pushing of tubule-like structures into the PEG space, which is really unusual because this is cross-linked PEG. It doesn't have any MMP binding sites to sort of allow cells to enter that space normally, but they do in the context of laminin 511. So I just share that. We can isolate those cells from the composites and expand them in culture, and the 411, especially in 511, result in pretty pure populations in a relatively short passage period. So by the second passage, they're almost exclusively CD31 positive cells. This also happens in 2D, which doesn't happen in the case of cardiomyogenesis. So these are just some images of laminin 111, 411, and 511, where at least half of the cells are CD31 positive. Again, just seeding iPS cells on top of these films. So something to think about if you're trying to develop a construct that wants to include or incorporate endothelium. We haven't completed functional analysis, so this is all couched in that. So I want to kind of end with one of the other sort of concepts that we're thinking about, and that is, you know, if we provide exogenous ECM on a film and a composite as a stimulus for differentiation, how do you consider or rationalize, you know, what ultimately happens in cardiomyogenesis, for example, where I think it's pretty clear that there are intermediates, right? And so how does that first signal actually translate into multiple signals? And so I've talked about how that second signal might be an evolving ECM that continues to provide signals 2 and 3. But what about that first signal? When you provide exogenous ECM, how does that actually begin to govern or trigger a differentiation outcome? And so our lab has begun to become interested in the intersection of engagement with an integrin via the extracellular matrix and other differentiation pathways. Here I'm showing Wnt, but it could be Notch or Hippo or others. We just really don't know a lot about that intersection. And so my group has become interested in one of the kinases associated with the focal adhesion complex, integrin-linked kinase, because it's been shown to at least associate with, and it's thought, block GSK3 beta. And so we took a look at our ECM stimulated cardiomyogenesis protocol, the one with the optimized formulation, and we looked at several days after exposure to ECM. So at day 1, you have sort of low localization of beta-catenin into the nucleus, but then at days 4 and 7, you see this increase and then maybe a gradual decrease here. Correspondingly, you see increase in phosphorylated ILK. And so this is just correlative for now, but if we then do some blocking studies where we look at OPT, which is our optimized ECM formulation, PEG only, which is no ECM, then our growth factor condition is the OPT plus growth factor, so that if the ECM pathway isn't triggered, we'll have the backup of growth factor. And then fibronectin, which is a ECM protein, but it doesn't seem to stimulate cardiomyogenesis. So if you look at beta-catenin, goes up only in the growth factor plus OPT or OPT situation. Phospho-ILK only goes up with the OPT formulation, and then if you inhibit ILK, you see that you pretty much annihilate the activation of beta-catenin except in the growth factor case, and similarly phospho-ILK. So we think we're starting to put together maybe a link between engagement of ECM and perhaps traditional differentiation pathways whereby we can inhibit GSK3 beta, maybe disrupt this complex and allow beta-catenin to transit to the nucleus. We're looking at or interested in how engagement of different integrins via different ECM sort of modulate the level of activation of ILK, sort of act as a rheostat that might further result in differential activation or inhibition of GSK3 beta or other proteins associated with differentiation pathways. So with that, I hope I've shared that the ECM is a strong stimulus of differentiation and maturation, and it's one of the principal pathways evoked with exposure at least of mesenchymal stem cells to ECM, and it's strongly tied to development. We want to investigate and hopefully encourage others to investigate the link between integrin engagement and differentiation. And we hope that all of this sort of builds into a gathering of data and information that can be used to physically localize ECM, either with 3D printing or other modalities, so that you can specifically induce differentiation without the addition of soluble factors, because if you're trying to develop something with multiple cell types, it's going to be hard to control where those soluble factors end up. So with that, just want to thank the students who did the work. They're highlighted in bold, and then Jay and Paul, who collaborated on much of this. Thank you.
So while Jack sets up, we have time for one question. Yes?